Good morning, everyone. Today I will be presenting an overview of the characteristics, habitat, virulence factors, and epidemiology of Staphylococcus haemolyticus. This presentation will give more insight into this gram-positive bacteria, which is part of the normal flora of humans and can cause opportunistic infections. Let's get started. Haemolyticus is a bacteria commonly found in our environment, on many human skin and mucous membranes. Though it is typically harmless, it can be an opportunistic pathogen, able to cause infection in people with weakened immune systems or those with medical devices in their bodies. Thus, its potential for harm should not be underestimated. Haemolyticus is a species of bacteria commonly found on the skin and mucous membranes of humans and animals. Particularly well adapted to aquatic habitats, it is often found in water, like the waves seen in the slide. Though it doesn't typically cause harm, it is wise to be aware of its presence and take steps to prevent it from spreading to vulnerable people or populations. Haemolyticus is a gram-positive bacterial cell that can be seen under a microscope. It is typically round and occurs in clusters, with a diameter of roughly 0.5 to 1.0 micrometers. This size difference is useful in distinguishing it from other microorganisms, and it also does not form spores, making identification easier. Staphylococcus haemolyticus is an important bacterium that can be encountered in clinical settings. Characterized by small colonies on blood agar that are typically white to gray in color, surrounded by a zone of beta hemolysis, this bacterium is also catalase positive and coagulase negative, making identification and monitoring important. Staphylococcus haemolyticus is a facultative anaerobe, capable of living without oxygen. It can grow on glucose and mannitol, and even more uniquely, it produces the enzyme coagulase, distinguishing it from other species of Staphylococci. Staphylococcus haemolyticus is primarily found to infect humans with indwelling medical devices, such as catheters, pacemakers, and prosthetic joints. It is important for people with these medical devices to be aware of this organism and to take appropriate precautions. Haemolyticus is a bacteria that possesses a range of virulence factors, allowing it to bind to host cells, medical devices, and degrade tissue. The presence of such features undoubtedly increases its ability to evade the immune system and should enforce caution in our efforts to limit the risk of infection. Haemolyticus is a bacteria notorious for being resistant to antibiotics. Often resistant to multiple antibiotics, it has acquired this resistance through plasmids or chromosomal mutations. Antibiotic resistance is an ever-present concern in the medical field, and Haemolyticus serves as an important reminder of this. Knowing the mechanisms of antibiotic resistance and how they apply to Haemolyticus is key in fighting this issue. Haemolyticus is a gram-positive bacterium that is found in hospital environments, often acquired through contaminated medical equipment. As a frequent cause of healthcare-associated infections and highly transmissible from person to person, it is essential that healthcare settings maintain the highest standards of hygiene to protect vulnerable patients. Haemolyticus is a dangerous bacteria that can cause significant infections in humans. It can colonize medical devices and damage host tissue, releasing enzymes that can break down our body's tissues. Additionally, it can form biofilms that protect it from our immune system, as well as antibiotics. This makes it a formidable foe that must be dealt with serious caution. Producing a strong immune response to Staphylococcus haemolyticus infection is a natural defense mechanism of the body. Antibodies are produced alongside the mobilization of specialized immune cells, allowing us to gain insight into the body's response. This understanding can help us refine the care and treatment we provide. Sample from the site of infection should be taken as the first step in diagnosing Haemolyticus. The sample should then be cultured on blood agar or other selective media. After identification using biochemical tests or molecular methods such as PCR, a definitive diagnosis can be made, allowing for the necessary treatment to begin. In order to stop S. haemolyticus from proliferating, good hygiene habits such as consistent hand washing and avoiding contact with those with potential or diagnosed infections should be implemented. Additionally, medical personnel must be careful to employ sterile practices during procedures and remove instruments including catheters, ventilators, and IV lines swiftly. Doing so can help limit the spread of infections due to S. haemolyticus. Staphylococcus haemolyticus infections can be treated with antibiotics such as vancomycin, daptomycin, and linezolid. However, antibiotic resistance should be taken into consideration when choosing the best course of treatment. Thank you for your attention.